Welcome back to A&P. I recorded our Zoom class the last time we met, and that is when we discussed the first half of chapter 26. So if you need to, go back and review that. Today we're going to be talking about chapter 26, section 6, which is all about making concentrated or dilute urine. Urine production is highly regulated, and the major hormone that does this is antidiuretic hormone, or ADH. The thing is, our body requires that we maintain our blood volumes within a specific limit, and therefore we're constantly regulating this, even if your fluid volume or your fluid intake is highly variable. So what happens is that when you intake a lot of fluids, you're not going to produce very much antidiuretic hormone. And this is going to result in dilute urine and a high volume of that urine. However, when you don't drink very much, this is going to result in a high level of ADH secretion, which causes a concentration of urine and a low volume of that urine. Glomerular filtrate is going to have the same osmolarity as your blood. So I want you to remember this number, 300 milliosmoles per liter. But the tubular osmolarity is going to change. So at the beginning, it's going to be 300, but these are going to change along the renal tubule due to the concentration gradient that's occurring in the renal medulla. When dilute urine is formed, you will have a low level of antidiuretic hormone. And this is going to change the osmolarity in the tubules. What you'll notice is that osmolarity increases in the descending limb of the loop of Henle. And that's because it's going into the medulla, which has a higher osmolarity. So it's going to match the osmolarity of the interstitial fluid in the medulla. And then it's actually going to decrease as it goes into the ascending limb, and then it's going to decrease even more in the collecting duct. And this is mostly because there is no antidiuretic hormone secretion when dilute urine is formed. The reason that osmolarity decreases as you go up or the ascending limb, even though it doesn't quite match the interstitial fluid, is because there's low permeability in that region of the ascending limb to water, but you have symporters that actively reabsorb sodium, potassium, and chloride ion. So that means it's going to leave water in the tubule, and the solutes are going to leave. So you can see that's going to make the osmolarity go down or have fewer solutes dissolved in the fluid. The thing about the collecting duct is this is what's going to be most influenced by the presence of ADH. And since there is very little ADH, the collecting duct will not be very permeable to water. So you won't lose water, and water will stay in the tubule, and that will make the urine dilute. When urine intake is low, you're going to want to make concentrated urine. So there's 4 different components that are involved in this process. First, symporters in the ascending limb cause the buildup of sodium and chloride in the renal medulla. We saw that before. We have these symporters, but what they do is they concentrate sodium and chloride ion in the interstitial fluid in the renal medulla. And that's going to help us with the next component in this process. The countercurrent flow mechanism through the descending and ascending loop of Henle is what's going to establish an osmotic gradient. And basically what this is called is the countercurrent multiplier. The way this works is that sodium and chloride that we looked at earlier is concentrated in the interstitial fluid. And then in the descending loop of Henle, water is highly reabsorbed in order to dilute that sodium and chloride. And that makes the concentration in the loop as it drops down be increased. And then more sodium and chloride are pushed out into the interstitial fluid, and then more water is pushed out in the descending loop in order to dilute it. And then you go to the ascending loop, it's more concentrated, so it's pushed out again. Now this is a little complicated, so I have a special video that I've included on Converge for you to watch so you can understand this a little bit better. The third mechanism that's involved in this process is that principal cells in the collecting duct are going to reabsorb more water because ADH is present. So again, let me remind you, when fluid intake is low, there's going to be a high level of ADH or antidiuretic hormone secreted, and that is going to cause more reabsorption of water. So basically, the collecting duct is going to become very permeable to water and allow for water to be reabsorbed, and this causes tubular fluid to become very concentrated. The final component of concentrated urine formation is urea recycling. Basically, urea is reabsorbed in the collecting duct and then secreted into the interstitial fluid and the loop of Henle. And this constant reabsorption secretion process causes a concentration of urea in the interstitial fluid, and it causes water to be reabsorbed to try and go and dilute it, which means you're concentrating the urine more and more and more as you're going down that collecting duct. We talked about what was happening in the nephron loop, but it's also important to look at what's happening in the capillary beds that feed them. The vasa recta is present in juxtamedullary nephrons, and again, these are the ones that are responsible for making concentrated urine. And they're going to dip into the renal medulla in a manner similar to the nephron loop. So this loop and duct together allows for the exchange of nutrients and oxygen between the blood and the urine. And what you're going to see is that the blood in that capillary bed is going to have similar osmolarity coming in and going out. So remember, around 300 milliosmolar. However, as it dips into the renal medulla, it's going to become more concentrated, very similar to what's happening in the loop of Henle. And so that countercurrent multiplier that we watched a video on helps to cause this concentration gradient that you'll see in the renal medulla, but this countercurrent exchange in the capillary beds are what's going to be responsible for maintaining that concentration gradient. So both are incredibly important for making more concentrated urine. So like I said, the vasa recta and the nephron loop are going to work together to help maintain the concentration gradient in the renal medulla, and that's what's going to be critical for making concentrated urine. So let's go back and review filtration, reabsorption, and secretion. Quickly, you have about 100 to 125 milliliters per minute of fluid that is filtered through the glomerulus, and that filters a lot of things, water and all the solutes in the blood except for basically blood cells. Then the fluid moves from the renal corpuscle to the proximal convoluted tubule. And the two processes we're going to see here are reabsorption and secretion. So we start to reabsorb water, and then we can reabsorb some ions such as sodium, potassium, glucose, amino acids. Secretion is going to be somewhat variable. You have hydrogen ions as well as urea and that sort of thing. But at the end of the proximal convoluted tubule, the fluid is pretty much the same concentration as what's in blood, or about 300 milliosmoles. You're going to have more reabsorption in the nephron loop. Most of your water reabsorption is actually going to occur in the descending loop, whereas the sodium and potassium reabsorption and chloride reabsorption are mostly going to occur in the ascending limb. You have some secretion as well, which means you're going to be dumping some urea into that portion. And at the end of that nephron loop, the tubular fluid is actually hypotonic, or less concentrated than what blood concentration is. So here it's about 100 to 150 milliosmoles per liter. At the early portion of the distal convoluted tubule, you're going to have a little bit more reabsorption of water, as well as a little bit of reabsorption of sodium and chloride via symporters. Finally, in the late distal convoluted tubule and the collecting duct, we're going to see more reabsorption of water. But this is only going to occur if ADH is present. If ADH is not present, you're not going to reabsorb water. You do have some secretion though, and the secretion that occurs is going to be of potassium and hydrogen ions. So what you see is that the tubular fluid leaving the collecting duct is dilute when ADH levels are low, and it's going to be concentrated when ADH levels are high, and that's because that's where all your reabsorption of water is going to occur. Okay, that's it for chapter 26. Let me remind you to go back and watch the Zoom video of our class on the first portion of chapter 26. Also, watch that countercurrent multiplier video and review the Chapter 26 handout and the reading guide and objectives so you know what to study for the quiz. There's a lot of other material available to you on Converge to help you study. And we will have a quiz on Friday, but it's also going to contain some of the Chapter 27 content. So keep up the good work. I'll see you soon.